The inner membrane of a mitochondrion of a cell has 4 electron transport complex such as complex 1, complex 2, complex 3 and complex 4. This inner mitochondrial membrane also contains F0F1 complex or ATP synthase. The complex 1 is NADH-CoQ reductase. The complex 2 is succinate-CoQ reductase. The complex 3 is CoQH2 cytochrome reductase. The complex 4 is cytochrome C oxidase. The green colored oval shaped structure present in intermembrane space is the cytochrome C. Every time it transports one electron from complex 3 to complex IV. In the mitochondrial matrix, the oxidizable substrates from various steps of reactions release electrons in presence of dehydrogenases to form NADH. The NADH is then converted by complex 1 to NAD+ with the release of a proton. This reaction also releases 2 electrons in complex 1. These 2 electrons flow to cofactor flavin mononucleotide or FMN. This FMN is related to FAD. Then these 2 electrons flow from FMN to iron sulfur cluster or protein. And finally, these 2 electrons flow from iron sulfur cluster to coenzyme Q or CoQ present in the inner mitochondrial membrane. It is also called as ubiquinone. It is a carrier of both electrons and protons. Each translocated electron causes a drop in potential of 360 MV, which is equivalent to delta G0 prime is equal to minus 16.6 kilocalorie per mole. So much of this released energy is used to transport 4 protons across inner mitochondrial membrane per molecule of NADH oxidized by complex 1. The addition of 2 electrons and 2 protons from mitochondrial matrix to coenzyme Q forms the fully reduced form dihydroubiquinone or CoQH2. CoQH2 releases its 2 electrons to another electron acceptor in the complex 3 called as cytochrome B. CoQH2 releases its proton into the intermembrane space or exoplasmic space and regenerates oxidized coenzyme Q. Within the complex 3, the 2 electrons are then transferred from cytochrome B to iron sulfur cluster and then to the cytochrome C1. Finally, the 2 electrons are transferred sequentially to 2 molecules of cytochrome C. For each pair of electrons transferred, 2 protons from CoQH2 and 2 protons from the mitochondrial matrix are translocated across inner mitochondrial membrane. This transfer of protons from CoQH2 involves proton motive Q cycle. The CoQ and cytochrome C both acts as mobile electron shuttles. They transfer electrons between complexes of electron transport chain. In complex IV, one electron is transferred from heme of each cytochrome C first to the copper ion called Cu(A2+), then to the heme in the cytochrome A, and next to the Cub2+ and heme in the cytochrome A3. Finally, the two electrons are passed to the ultimate electron acceptor O2. After accepting the two electrons, 0.5 O2 or half O2 or an oxygen atom and two protons combine to form one H2O molecule. During transport of two electrons in complex IV, two protons from mitochondrial matrix are translocated across the inner mitochondrial membrane and reach to the intermembrane space. The mechanism by which the protons are translocated is unknown. Now, let us calculate the number of protons translocated from mitochondrial matrix to the intermembrane space through the inner mitochondrial membrane per pair of electrons that flow from NADH to O2. Through complex 1, 4 protons are translocated. Through complex 3, 4 protons are translocated. Through complex IV, 2 protons are translocated. So, the number of total protons translocated are 10. Now, let us discuss what happens to the FADH2 electrons in the ETS. During the TCA cycle, succinate dehydrogenase oxidizes succinate to fumarate with the release of 2 protons and 2 electrons. This reaction generates FADH2. This succinate dehydrogenase is one of the 4 subunits of complex 2. In this way, TCA cycle is functionally and physically linked to electron transport chain. The 2 electrons released in the conversion of succinate to fumarate are translocated first to FAD in succinate dehydrogenase, then to the iron sulfur cluster, and finally to the coenzyme Q or COQ. The coenzyme Q binds to a cleft on the matrix side of the transmembrane portion of complex 2. Coenzyme Q then combines with 2 protons from mitochondrial matrix and with 2 electrons to form COQH2. The COQH2 then regenerates coenzyme Q with the release of 2 electrons to the cytochrome B in complex 3 and 2 protons to the intermembrane space. Then these 2 electrons flow from cytochrome B to iron sulfur cluster and then to cytochrome C1. Finally, 2 electrons are transferred sequentially to 2 molecules of cytochrome C. For each pair of electron transferred, 2 protons from COQH2 and 2 protons from mitochondrial matrix are translocated across inner mitochondrial membrane. This transfer of protons from COQH2 involves proton motive Q cycle. In complex IV, first 1 electron is transferred from heme of each cytochrome C to the copper ion called Cu(A2+), then to the heme in cytochrome A, and next to the Cub2+ and heme in the cytochrome A3. Finally, 2 electrons are passed to O2. After accepting 2 electrons, 0.5 or half O2 and 2 protons combine to form one H2O molecule. During the transport of 2 electrons in complex IV, 2 protons from the mitochondrial matrix are translocated across the inner mitochondrial membrane and reach to the intermembrane space. The mechanism by which the protons are translocated is unknown. Now let us calculate the number of protons translocated from mitochondrial matrix to the intermembrane space through inner mitochondrial membrane per pair of electrons that flow from succinate to O2. Through complex 2, no protons are translocated. Through complex 3, 4 protons are translocated. Through complex IV, 2 protons are translocated. So the total number of protons translocated are 6. The mitochondrial membrane is impermeable to protons. These protons cannot diffuse back into the matrix across the membrane, but these protons can enter the membrane via membrane-bound ATP synthase or F0-F1 complex. It is a multi-enzyme complex containing 2 parts like F0 and F1. This F1 acts as ATP synthase. This multi-enzyme complex forms a proton channel through which 3 protons flow from F0 to F1 and reach to the mitochondrial matrix. This backflow of protons down the proton gradient provide energy for ATP synthesis, in which ADP and inorganic phosphate or PI forms ATP. This process of synthesis of ATP at the expense of proton motive force is called as chemiosmosis. Please like, subscribe and share.